Hello, my name is Xiaoqiong Wei. I'm so happy to present my work about detecting gene ontology misannotations using taxon-specific racial analysis. Okay, sorry. Firstly, I want to introduce myself. I graduated from Sichuan University from 2015 to 2016. I was a visiting PhD student in Yangzhang Lab. Before joining Yangzhang Lab, I never know how to write script. It is Chengxin who helped me much more on script writing. So that is why he become my husband. Here, I want to give my suggestion to those single boys. So if you want to have a wife, please learn to program well. This picture is taken in my wedding. My PhD mentors, Dr. Zhong Li and Dr. Yang Zhang. They signed on my marriage license. It means that they are both responsible for my marriage. Anyway, the knowledge I learned in Yangzhang Lab helped me get a postdoc position in Lingqi Lab. So this is my current postdoc lab. Dr. Lin Qi, who sits in the center of this site. So I joined this lab in March 2020. So it is the beginning time of pandemic. So we have to wear mask even now. Pandemic changed my life. I lost my dear grandma. At the same time, I get a baby. So he gave me a lot of joy and work. In this March, I came back to work as a nursing mom. So now I am learning how to balance my family and work. Okay, it's a long self-introduction. Thank you for your time. Let's talk about science. In this project, we developed a taxon-based ratio method to detect GO misannotations. Here we also point out where the misannotations come from. What is a GO? GO is a gene ontology, which includes the molecular function, biological process, and the cellular component. We use protein function in public database, such as UniProt, especially when we get next generation sequencing data, we will use GO annotations to do function enrichment analysis. So here is an example. In this paper, they performed single cell RNA sequencing from two kinds of parasites. After get differential genes, they did GO enrichment to analyze the different gene function between these two parasites. So protein function annotation is very important, but not all of them are validated by experiments. So it means that some of them are not right. Here we can see that most proteins lack manual function annotation. The upper line is the UniProt. The upper line shows the number of protein sequences in UniProt database. In 2020, there are more than 180 million protein sequences. However, in the Swiss Prot database, the lower line, the Swiss Prot database is a database with the proteins annotated by the experiments. Those proteins have the manual function annotation. We can see that there is only 560 million proteins were annotated by experiments. It means that there are more than 99% of proteins are not experimentally characterized. Here we can here this is a statistics image image from the UniProt database. We can see that in all organisms. The IEA evidence code has the 99%. So IEA is a short name of inferred from electronic annotations. It means that the 99% GO terms are computationally predicted without manual review. So even though in 20 model organisms which are well studied, the IEA percentage is 54%. These 20 model organisms include the bacteria, include the human, mouse, E. coli. So we can see that even though in this well-studied models, there are still 54% proteins without experiment validation. Here is an example. This is a human protein, junction globin. This protein is annotated in the UniProt as the structural constant of the cell wall. Similarly, in Nextprot database, which is a subset of the UniProt database, we can see that they also annotated this protein as the structural constant of our cell wall. We know that no cell wall in human cells, so it is an obvious error. We check this experiment paper and found that this paper only show that this protein is on the outer surface of the cell. So somehow the curator incorrectly assumed that this complex is the cell wall, resulting in this misannotation. So this protein is well characterized, but also contain the errors. So we decided to develop our own method to find out its errors. So here is the principle of our method. Firstly, we classify proteins based on their taxon. In this example, we classify the proteins into 4 taxons in 4 columns: animal, bacteria, fungi, and plants. So the column shows the GO terms. For each protein function, we count the portion of the proteins in each taxon annotated to this function, which is shown here as the area of rectangles. We ignore its functions if they are annotated to about the same amount of the proteins in all taxons. So for the upper 2 rows, we ignore this kind of protein. So the next two rows, we can see that this protein, this GO term is only present in one taxons. So we also ignore this kind of proteins. For the last 2 rows, we can see that the number of proteins in the bacteria is much less than other taxons. Similarly, for this GO term, the number of proteins assigned to this GO term is less than the fungi. So it means that these 2 GO terms are likely to be misannotated. Here, this is the equation of our method. So for a function Q, its annotation rate in taxon T, we called the PTQ, equals to the number of the proteins annotated with the Q in this taxon, divided by all the proteins in this taxon. Then we calculate the ratio. Equals to the minimum of PTQ divided by the maximum of the PTQ. Then we, what's happening? Then we select 20. We select 20 model organisms, including the 5 animals, 7 bacteria, 3 archaea, 3 fungi, and the 2 plants to apply our retrieval analysis to flag potential misannotations. After manually checked the flagged annotations, we found that about 200 misannotations for each UniProt annotation release. So 200 misannotations does not sound a lot, but remember that this is only for well-studied model organisms, such as human, mouse, and E. coli. So actual the amount of misannotations for other less studied organisms is much larger. We also check which year the misannotations was made in UniProt, as shown in the x-axis. Here, the black and green colors are the RIR analysis using the different taxa. We can see that more than half of the misannotations are made in UniProt on 2017 or earlier, and was not corrected for more than 3 years by the time we published our study. We next analyzed what kind of evidence codes in these misannotations. For the 20 model organisms we picked, we found that most of misannotations have IBA evidence code. IBA is the short name of the inferred from biological ancestor. It is a semi-manual computational function prediction based on the phylogenetic tree. To understand why IBA geoterms are so easily misannotated, we checked some examples. Here, UDG is is a well-studied enzyme for repairing DNA. Bacillus is a kind of bacteria. So in Bacillus, this protein has served a structure, which I showed on the right. Despite being so well characterized protein, its UniProt annotation assign the function, assign this protein as the mitochondria and the nucleus, both of which are not in the Bacillus bacteria. We can see that in this database, which is a Bacillus-specific database, we can see that they also assign this protein as the nucleus and the mitochondria. So we know that in the bacteria there is no mitochondria and nucleus. So this is also an error. So next, we want to know the principle of the IBA method, and also we want to know why the IBA IBA misannotated these proteins. So here is the principle of the IBA method. We can see that for these 4, 5, 6, 7 are the proteins, 1, 2, 3 with a circle are the ancestor. So the 4 and 6 with the green green square are the proteins with known function. 5 and 7 proteins, we want to know the function. For now, we don't know. So we can see that for the 4 and 6, the most recent common ancestor is 2. So for the IBA method, they will assign the protein 5 as the same GO term with 4 and 6. Here is the reason why the UDG is assigned to the mitochondria. So we can see that this is the phylogenetic tree of the UDG family. The first branch includes the animal, fungi, and the lower plants. The lower branch includes the bacteria. So we can see that for this lower branch, there is a small branch which included in the upper branch. So for all these 4 branches, the UDG proteins are assigned to the mitochondria and the nucleus. So so we can see that the most recent common ancestor for these 4 branches is here. So this is a best area we want to study because they have the most recent common ancestor. So the UniProt assigned the mitochondria and the nucleus to the UDG protein in the base layer. So this is the reason. This is the reason why the IBM make this mistake. Actually, the UniProt and the GO constantly correct misannotations for the 20 species we collect in this study. There is 4.5% of geoterms annotations originally presented in UniProt-GOA release 2018, which were removed in the 2019. So among its corrected geoterm annotations, 81% has the IEA evidence code. 13% of proteins have the IBA evidence code. Using our IR method, we found 250 misannotations. Among its 250 misannotations, there are 63 misannotations were corrected in the 2019 version. Among these 63 potential misannotations, 47 have the IBA. 47 have the IBA evidence code. So this data suggests that our IR method indeed detect potential misannotations, especially those have the IBA evidence code. As an additional sensors of the quality of IBA versus other types of geoterms, we collect 10,500 geo annotations from 2018 that are either rejected or confirmed experimentally in 2019 version. Among these annotations, there are 1,500 IBA terms. So it's the percentage is 31%. It means that there are 31% of which are rejected in the 2019. This is an estimated error rate 6 times greater than fully automatically annotated IEA terms. IEA terms only show the 5%. And we have report our findings to UniProt, the GO, custom, and the next prot. So most of the, for now, most of the misannotations were corrected. So here I want to show the key findings. So firstly, we developed a semi-annotated approach to detect misannotated functions. And also there are 53% of these misannotations in the UniProt for larger than 1 year. And also we we reported that 66% of misannotations are from the IBA evidence, and also we also clarify that the reason why the IBA misannotated these proteins because they use a phylogenetic tree. I want to mention that we use the ITESA cofactor to predict a protein function for uncharacterized proteins. So this method includes 3 parts: protein sequence alignment, protein-protein interaction prediction, and the protein structure prediction. For example, for this protein UPE1 protein sequence, after we get this protein sequence, we will do the site-blast to get the sequence co-homologs. And the second part is that we will use the string database to analyze protein-protein interactions. The third part is that we use ITESA to predict the structure, to predict this protein structure, and then we will do structural alignment. Then we will do structural alignment to get the structure analogs. So finally, we will give different weights for these 3 parts, and then run the cofactor to finally get the protein function. By the way, I want to mention that using ITESA, we predict the structure of the nephrin, which is an important protein for the kidney function. So this work is published and will be present in the April issue of GCI as a cover story. Thank you, Dr. Yang. Dr. Yang Zhang, I learned the structural analysis in that lab. Thank you. That's all my presentations. Welcome questions.
If anyone has any questions, you can either unmute yourself and ask, or put them in the chat box and I can read them to Xiaoqiong.
Yeah, welcome questions.
So Xiaoqiong, it's very nice presentation. And it's Gil Omen. It's nice to see you develop this whole area of research and to see the application with the Kretzler lab on the kidney. Can you explain to us a little more what your interactions were with UniProt and NextProt, and what it takes to get the international databases like those to hear your criticisms and findings and actually take action rather expeditiously, promptly? Tell us how that worked. Do you understand what I'm asking? What was the process of informing UniProt and NextProt of your findings of these misannotations?
Oh yeah, for the you mean that for the misannotations, what we do in the UniProt, right?
Yes. Do they have an office for complaints or adverse findings, or did you have to find someone to take responsibility there? How did it work?
Yeah. We emailed UniProt Dr. Lady Lane. Oh no, this is the NextProt boss. We send our misannotations proteins to the UniProt, NextProt, and the GO consortium. They said that they will check them and correct them. Especially for the NextProt, Dr. Lady Lane. She is the boss of the NextProt. She has checked all of the misannotations and corrected them. Also, she downgraded the IBA evidence from the gold to silver. We also checked the proteins I show here, which misannotated. For now, in UniProt, NextProt, they both correct these misannotations.
Great.
For now.
Well, I would suggest that you send a copy, maybe a compressed copy of your slide set to Dr. Lane and to others, and let them see how much you've accomplished in this area. It's really terrific.
Oh, yeah.
And it's good to know that those big organizations respond to such analysis.
Yeah, thank you. Yes. Any other questions?
So far, nobody's entering anything in the chat box, but again, if anyone has questions, you can enter them in the chat box or unmute yourself. Xiaoqiong, this isn't a question, but Dana just wrote in there, no question, but great talk, and just thank you for your great talk. So I just want to pass that along to you.
Oh, thank you.
Well, if there are no questions, then I'd like to thank
comment.
Xiaoqiong for the excellent presentation today. Hopefully everyone can join us next week for the last seminar of the semester. We'll be hearing about we'll be hearing from Abhinav Atreya about identification of collateral lethal metabolic targets for precision medicine and cancer. So thanks everyone for joining us today.